Hello, everyone. I'm Yukun Tan, a postdoc from the University of Texas MD Anderson Cancer Center. Today, I would like to present the novoBreak-rna, the local assembly tool for novel splice junction and fusion transcript detection from RNA-seq data. So why is it important? As we know, the fusion transcripts often have an important role in the initial steps of tumor genesis, and the reviews from have estimated that gene fusions occur in all malignancies. And it potentially can be used for cancer diagnosis, prognosis, and therapy. Also, we know alternative splicing, the dysregulation of the alternative splicing occurring in every one of the hallmarks of cancer, and different types of alternative splicing affects the resulting translated protein products. In addition, the alternative splicing of the cancer-related genes can affect cell cycle control, signal transduction pathway, and so on. So next, I will introduce the schematic diagram of the splice junction and fusion transcript. So as we know, when a gene is transcribed, we actually first have a pre-mRNA products and then take the exon sequences and forming them together to produce the mature mRNA. So what we remove here, we call it junction. Then based on the location of the junction, if the junction is located at the single gene, like the figure 1, so it means that it is the process of the selecting different combination of the exon. It is called the alternative splicing. And each kind of combination is isoform. And if we detect the removed junction like here between two genes, meaning that it's a fusion transcript which caused by the genomic rearrangements. So nowadays, a lot of tools can detect such junctions, but they either based on alignment-based approach or the de novo whole transcriptome assembly approach. Alignment approach means directly align the short-read RNA to a reference genome or align to a reference transcriptome. It is computationally efficient, but limited in detecting sequences when it is unmapped to reference. For the assembly approach, the approach will be the short reads can fill in gaps not covered by long inserts, but it's computationally expensive and potentially limited sensitivity when the sequence is low, when it's low sequence coverage. So in this study, we proposed a local assembly-based framework called novoBreak-rna, which modifies our well-attested genomic structural variation breakpoint assembly tool called novoBreak. So there are 4 steps. The first one is decompose the RNA-seq reads and reference sequencing into k-mers. Then we identify the novel k-mers. So what means novel k-mers? Novel k-mer means not existing reference, for example, the ref-seq, ensemble, or gene code reference, but also present uniquely in the tumor RNA-seq reads. And then in third step, we cluster the novel k-mers and do the local assembly. The last step, we align the longer contigs which assembled in the previous step and map to the genome reference. So the output of our workflow will be the novel junctions. The advantage of our workflow will be more sensitive in assembling novel sequence junctions and more efficient than the whole transcriptome assembly. So after we got the novel junctions, we designed several general filters. For example, we identifies and removes all the duplicate reads induced by the PCR by using the P card. And we also consider the anchor length. So we will remove all the junctions with anchor length lower than a threshold, for example, 10 BP. And also we control the mapping quality. And also we consider the number of supported split reads, and also the proportion of the split reads and the sequence depth of the coverage. And there are still some filters designed specifically to the fusion transcript For example, we remove the candidate fusions with high number of reads on pseudogenes, and also high expressed genes that are likely to be involved in fusions. So next here is the results. So we first generate the synthetic data based on the different splice events located on the chromosome 21. And the reference here means the ground truth of the novel junctions. And we can see our tool have a higher sensitivity than the other. And we also apply our tool to the real data of the breast cancer. Here are 4 cell lines of the breast cancer and 26 validated fusion genes. We can see our tool detect the highest total number of the validated fusion genes. So how to explain this? So the most important reason is that we fully use the unmapped reads. For example, for these 4 cell lines, the total number of reads are around 200 millions, and more than 7 million reads are unmapped. So if we only use the unmapped reads, after assembled, we will get more than 100,000 high quality contigs. And we can, based on these contigs, we can find 8 true fusion transcripts. And 5 of them pass all the filters we designed. And more interesting is that 2 of them don't have support from any mapped short reads. So that means theoretically, these 2 fusion events will not be detected by the alignment-based method. So that's all for our tool. So at the last, I would like to thank all my co-authors for their great help. Thank you.